Hi, my name is John Lees. I'm from the European Bioinformatics Institute in Cambridge. I'd like to start by apologising for the absolute mouthful that I've named this talk. Really what I'm going to be telling you about are a few different bioinformatic methods and bits of software for analysing pneumococcal genomes that we've been developing over the past few years. Most of the talk I'll focus on the first two, which are for genomic epidemiology, specifically clustering and subclustering pneumococcal sequences into strains, but I'll also end on pointing out some web tools you might find interesting as well. I've got a few videos of these working because I'm not brave enough to use them live. But overall, what we've been trying to do in the past few years is develop tools which work with surveillance data, so particularly genomes produced in real time, or a lot of them, and continuously, helping people integrate queries either from local studies or their own collections or things of interest into a more global context, so using all of that data. We want to develop tools which sort of give you the basics, so the strain, which I'll define in a minute, serotype and AMR prediction. And we also want to develop tools which work both for bioinformaticians and non-bioinformaticians. So to motivate the first part, as we know, Streptococcus pneumonia has many strains. So here's the kind of cartoon picture of a tree. So within a strain, you have samples or genomes which are close together. You can draw a tree and do lots of interesting things within there, but between different strains, they are distantly separated, almost as distant as species separation, but not quite as distant as that. Here's a real picture of a tree. So this is a whole genome phylogeny of about 600 genomes from Massachusetts, from this paper by Nick Croucher from 2013. What you can see here is that sort of towards the tips here, you have these clusters, which there's obviously some structure in and you can do things with and actually construct quite a good tree, more amenable to analysis. But the center of this, in fact, actually it's not shown that well on the phylogeny, is very uncertain, and the ordering of these branches is impossible to resolve with this kind of data. So we probably just shouldn't bother trying to resolve it. And what would be nice to do would be able to find these clusters in large data sets, find useful clusters which you can then do sub-analysis on. So this is exactly what a typing scheme is for. And what we want to do in developing a typing scheme is we want to avoid doing whole genome alignments, and we want to avoid doing large phylogenies, which will not scale to surveillance data and have other problems too. So why do we develop typing schemes? Well, we wanted to split population into clusters or strains. We also want to make sure that what makes it different from clustering and typing is that those clusters will have the same names. We make sure we're talking about the same thing. That lets us look at other properties of that same strain or that same type, do phenotype association by proxy, and overall it needs to represent vertical inheritance. And then sort of at an engineering level, we also want to do this while using all of the genomic diversity in the species to get the best resolution, so including accessory diversity. We want the clusters to be useful and biologically meaningful. We want to be able to define subclusters, so as well as the top level clusters, do subclustering within that. It's useful for lots of purposes. We need to be robust to sequence errors, because as we have more data, we definitely will have those, and annotation errors. So gene by gene approaches aren't very good for that. And we also need to scale to hundreds of thousands of genomes. So there are methods like MLST, CC, and so on. However, they don't sort of fulfill all of these requirements. So this led us to develop the method which we call PopPUNK, which does population clustering based on K-mer distances. So it's alignment free, so it's very quick. So you can very rapidly create a plot that looks something like this, which gives you the core and accessory distance. This is a sort of tens of thousands of pneumo, and you can optimize a model which picks basically just a boundary in there. It picks a cutoff, which gives us good clusters. So I'm not going to actually go into much more detail there. I'll give a talk at ISPPT in more detail about how that all works and how we optimized it. If you're not going to be at ISPPT, I will also try and put that on YouTube afterwards. I'd also like to distinguish PopPUNK, which is the bit of software and the general method, from the particular clusters or strains that result from that, which are Global Pneumococcal Sequencing Clusters, or GPSCs for short. These are numbered from 1 upwards. roughly, although not exactly. The smaller ones tend to be larger, so GPSC1 would be the biggest one. At the moment, we're on version 5, which has about 42,000 genomes, about 1,000 GPSCs, which vary in size. For those of you familiar with gene-by-gene approaches, the resolution is similar to CCC, to clonal complex, although there's lots of advantages, kind of based on the last slide. But even when you look at the clusters, you see that the GPSC ones looks better. If you're interested in this in more detail, have a look at Narendra Kumar's poster. Another advantage of getting these GPSCs is Rebecca Gladstone has created a big collection of resources. So for all of the common GPSCs, you can look and find out, you can get the trees of them, you can see how they were sampled globally, also over time. You can look at serotype information, you can look at AMR, you can look at recombination, all interactively in the browser. So once you have one, it's a very useful bit of information. So now I've convinced you that they are useful. How do you actually get them? If you want to assign a lot of genomes to GPSCs, use the command line. We've got instructions on the Pneumogen website. Or if you just want to have a look or do it one by one, you can also do it interactively in the browser by Pathogen Watch. It's a little bit hidden, but you can see it's down here. So here I've filtered GPSC1 and I can see information about those. So great, that's kind of done the first bit of task here. It's split this up without needing to use phylogeny, without needing to use big multiple sequence alignments. We can now actually do some of this analysis on these subcomponents. So we want to automate some of this. Again, I'm not going to go into the details of all the tools, but we've kind of picked some efficient tools here. And what we can do is we can automate all of the different subcluster analysis so we don't have to run each cluster one by one. And again, the principle is that we don't make a tree of the whole species, but where we can, we do make high resolution trees and kind of stitch all of that together. You end up with this kind of quite complicated graph. In practice, it's quite simple. You just run snakemake. You can pick a target. So this will actually also make the visualization for me, the -J32. I'm just setting the number of cores. So for about 600 genomes on my computer, this took about 6 minutes, didn't really use much memory. When it runs, it will automatically work out what it needs to do. It'll submit all these jobs. It'll give you a summary and then it will run through them all. And then at the end, I just get a URL. Click on that URL. This is a link to a Microreact. If I play that, you can see me playing with it here. So here I've got the whole tree. I mean, sort of drawn in a slightly cheaty way to avoid all of the computation I mentioned before, but then we can sort of zoom into individual subclusters. So we can look at strains, but then you can see we've got subcluster designations within there that reflect what's in the tree. We've got a more accurate tree for the whole subcluster. On the left there, we've got accessory genome content. On the right, it's based on core genome content, so we can see some structure within there, so probably different genes, which we could look at in more detail. We're going to keep adding to this. There's a few things still needs to. We're going to add the recombination detection and removal, similar to what Rebecca had for the individual GPSCs. We'll also make transmission reconstruction, and we'll sort of make this so you don't have to use the command line at all. In the spirit of making web tools, I'm going to tell you about two web tools just kind of briefly at the end, which also sort of fill in some of those aims at the start. So we've got strain prediction. Serotype prediction is ongoing. I'm not going to show you that, but AMR prediction, we developed this method which is actually quite general. It's not specific to the mechanisms in Streptococcus pneumoniae. Basically how it works is like this. We fit quite a simple machine learning model, specifically the elastic net, just to sequence content. So small sequence words like k-mers called unitigs. And the nice thing about this is that the result is just a linear model. So it's simple to run, it's simple to analyze. We genuinely held out some data to our fit so we can look at how this performs in other data sets. And generally for these 5 drugs, it does a pretty decent job. So sort of about between 85 and 95% balanced accuracy. So certainly good enough for a quick look, not enough for anything too serious. You can run it here. One of the cool things about it is that it all runs in your browser. This is just called WebAssembly code. So everything is just run on your own machine. You don't have to install anything either. I'm just going to show here loading the example sequences, but you can drag and drop. But this means you don't have to send any genome data over the network, which can be slow. If it's sensitive, you don't have to do that. Nothing is being sent to us. The analysis is all being done on your own machine. You can hover over, get some sort of more information, and we've got guides about how to interpret that. And then finally, I just want to briefly tell you about this tool called BacQuerya.com, just as a bit of an advert. So this is in development, but we've got about 12,000 pneumos in there and some E. coli. And basically what this is this is a search engine, but for bacterial genomes. Particularly, it was to solve some of these questions. So you see a study and you want the genomes from it. How do you actually get them to reanalyze them? If you have a gene of interest, perhaps potentially interesting vaccine candidate, and you want to know whether it's conserved, what the population variation looks like, you can search for that gene. It'll make it easier. Also, you can run something that looks a bit like a BLAST, but not just through the RAFC genomes. So again, I'll show you a quick example of that here. So I searched my query. I've actually misspelled Streptococcus on purpose here, but I'm going to look for certain GPSC, GPSC 9, I think, that comes from Nepal. So I just put my query in. I can do some filtering, get my search results back pretty quickly. I can look through information about those that's pulled from the ANA and other sources. I can look at whether it's contaminated, whether it's good assembly. You can also see what genes are in there. Does it have TET-M? It does. And then I can link. I could have searched for TET-M, but here I'm just linking straight through to it. I can see TET-M. I can get subsampled alignment. This is kind of good to see sort of how much variation there is in there. You can see there's quite a bit in the middle. And then that also links back to samples as well. So just to end there, I'll summarize the different bits. So GPSCs, this is a consistent top-level nomenclature for strains in Streptococcus pneumoniae. You can get lots of more information once you know the GPSC. You can assign them to new genomes using PopPUNK on the command line, Pathogen Watch on the web, or Bebop, which is sort of a full web-based interface, which is coming soon. You can work within those GPSCs to do sub-analyses such as transmission analysis, recombination analysis. You can use our PopPIPE pipeline to automate all of that and do that on large data sets much more easily. You can get reasonable AMR predictions in your browser, and I hope they'll get better. And we want to put all of this information together. That is hard, but I think it's getting better. Our attempt at doing that is BacQuerya. So I'm there, thank these people, particularly Nick, who I worked on PopPUNK with, Marie, who developed AMIMA, and Steph and Miranda, who've done a lot of the hard work with GPSCs. And I'll just end with my summary list of software there. Also, if any of that is interesting, you can find it listed on my website.